Okay, welcome to this first webinar of World Federation of Parasitologists, the road to ICOPA 2026. Welcome both to those who are joining live right now and to those who are watching this afterwards. My name is Pikka Jokelainen. I'm the president of the World Federation of Parasitologists. And this webinar series, the Road to ICOPA 2026, a brand new initiative from our executive board. So this is the really first thing in this series and new things happening in our work. I will be starting by a brief overview of World Federation of Parasitologists and ICOPAs, and then we will go to the theme of this webinar and look at One Health, both the definition and how we can work with it from the parasitology point of view, or how we can use One Health point of view to parasitology. It goes both ways. And then I will have two examples, very general, on some thoughts on One Health surveillance, and another one on a specific project, One Health collaboration on source attribution of zoonotic parasites. And then we will stop the recording, and those of us who are joining live, we can stay on this platform and discuss. First, I want to say that I am here mainly in the role of the president of the World Federation of Parasitologists, so it's really nice to be opening this webinar series and our work towards and road together towards ICOPA 2026 by these webinars. On my daily work, I have a background in doing a lot of research and work with parasites, different parasites, mainly zoonotic parasites, and using the One Health approach. My daily work is in Denmark, Copenhagen, in this building shown in the picture, so State and Serum Institute. Our organization is a public health institute, and we also have veterinary preparedness functions in collaboration with University of Copenhagen. So we host reference labs for both human pathogens and animal pathogens. So for example, for many parasites, we provide and do tests for both human samples and animal samples, and also some environmental samples. And of course, the work is a lot about multidisciplinary research and collaborations. I am especially involved in international collaborations in the field of One Health. Interested in systems and structures and all that, and always reflecting on what we can learn from different point of views in our work. I would also highlight this, kind of like everybody who is joining, to think from your own background. Where are you? How is your work linked to the One Health concept, and how it maybe has been developing over time? This webinar series, the Road to ICOPA 2026. So we are on our way to Montreal, Canada, where the 16th ICOPA, so 16th International Congress of Parasitology, will be taking place in August this year. And this slide here also highlights that the registration is now open. So our series start of these webinars is very timely. And I would like to highlight that I'm actually wearing the same dress as in this photo on the slide. And we will come back to that flag. That is the flag of the World Federation of Parasitologists. And I have been holding it in the past. Now it is actually already in Montreal, Canada, waiting for us all. Very briefly about the World Federation of Parasitologists. This description is from our website, highlighting that what is the aim of our structure is to promote and coordinate exchange of knowledge, research, and other activities relating to parasitology. We are an umbrella organization, so the members of our organization are scientific organizations, local and regional ones that are concerned with parasitology and parasitism. So both national, regional parasitology, some tropical medicine. It's quite wide scope also in that. We are already have quite some history behind us. The Federation was founded in Poland in 1960. And a key activity of the World Federation of Parasitologists is organizing ICOPA, the International Congress of Parasitology, every 4th year. And this is pictures of the members of the current executive board. I would like to thank them all for our work together and also for this idea and enthusiasm and ideas for this webinar series. Really nice to work across the globe with people with shared interests in parasitology, and at the same time really complementary and different approaches and expertise areas and so on. So it's the diversity is clear, and it's really a strength and wonderful to work together. Also, I think exemplifies what parasitology at the global scale is. It is working together. And that is, in my mind, very much aligned with the One Health principle. ICOPA, our Congress taking place every 4th year. Here's a list of all the ICOPAs since the start of them. And if you think of ICOPAs or the life cycle of ICOPAs, it's indeed there's a pre-patent period of 4 years. And then the fun is for 1 week. The patent period is about 1 week, the Congress. And mission is happening by passing the flag that we saw already earlier. These different stages have taken place in different places of the world. However, I can see there are still some gaps, some corners of the world we have not yet been having our ICOPAs, but it is a global effort. The hosts of each ICOPA, they are local and regional parasitological societies. And now in this kind of parasitology talk, I maybe need to highlight that ICOPA is a very nice parasite, so it doesn't harm the hosts. And hosting ICOPAs is very attractive and highly competed to get to be the host. And I was myself very happy to be the chair of the ICOPA 2022 in Copenhagen, here in Denmark. It was organized by the Scandinavian Baltic Society and the Danish Society for Parasitology in collaboration. And the selected host has a lot of work, but it is also a lot of fun. The host needs to provide a good venue for the ICOPA to develop and flourish. And I mean, that's the patent period. That's the most important part of the life cycle. And also there happens the transmission ceremony to the next hosts. And the flag is a key item in this ceremony. And here you can see 2 photos actually of me holding that flag. So the flag of World Federation of Parasitologists is passed on from the previous host to the new ones. So in the first picture, this is from South Korea from 2018, where I was receiving this flag. And then the picture used in the advertisements of the upcoming ICOPA 2026 is actually from Copenhagen ICOPA 2022. This is from the opening ceremony, where the flag is taken to the stage. And then at the end of the Congress, the flag was given forward by me to the next host, so colleagues from Canada. And now the flag is waiting for us, indeed, there in Montreal. But this is the life cycle. I think challenge is that ICOPA is only once in 4 years. And that's why it's very nice that we also have some activities here in between ICOPAs. And this webinar series is one of those that we are now introducing. Then to the topic of today, One Health approach. It is a topic that many people who work with One Health say we've been doing it longer than we actually use the word, because it is kind of core of it all. It makes a lot of sense. At the same time, we are very happy that a few years back, there was work done to come up with the definition of One Health. And this definition by the One Health high-level expert panel, there's a publication on it where there's the whole text, the description, and then there's also this illustrative image about what is One Health. And the key thing is that it's an integrated, it's unifying approach, and it really is an understanding that the health of us humans, health of animals, and our common environments, they are closely linked and interdependent also. And typically working in One Health way is very much in like between sectors or across sectors. There's different disciplines, expertise areas that need to be together. That's both challenging and really rewarding. Sometimes we notice that we need to work some time together to have a common language and understanding, and then we can really help each other to in the topic we are working on. In this picture, in the center low part, you can see the classical kind of humans, animals, environmental here shown as ecosystems. That is the kind of the three things that are highlighted in many classical definitions of One Health. At the same time, this definition also expands to wider things like the society, how there are different disciplines and sectors, and also the kind of the key functionalities across. I have noticed that in my daily work and my research work, if it's on zoonotic parasites, it's usually very specific, certain host species, maybe environmental stage is relevant in the considerations, and it's like really in a part of this. And overall, in my daily work here, like we are working in infectious disease preparedness, so it's the humans, animals, a little bit environment, focus on the pathogens that could go across these compartments. And I work a lot with systems for surveillance and preparedness. So that's usually there are these different components at least discussed. And then I've noticed more and more in my One Health leadership roles that it's a lot about those structures that are there in the upper part of this image, the collaboration, capacity building, coordination, and communication, and how to make sure that we can work together when One Health brings us together, and make sure that that is smooth and successful. When I look at this picture, I usually have this on my One Health presentations. I always notice something new in it. And I've been asking also in some of my presentations if the audience can find something surprising in it. I encourage you all to think from that point of view. I can share my observations. One thing is the healthy humans. This work was done during the COVID-19 pandemic, and how the healthy humans are shown in this illustration is actually like you see me. So only the upper part of the body. So perhaps they are also in a webinar in this. So it's very healthy to be here in this webinar for sure. But that is kind of funny small thing that it tells about the time when this work was done. So that's an interesting part of the humans shown in this picture. Then the ecosystems part, the environment is shown with quite macroscopic things. There's like mountains and trees, and that kind of makes me think like, how about all the microbiology and microbiome and all that's like, that's also where things are interconnected and affecting each other. So somehow I feel like something is lacking. Of course, that is microscopically small, so fair enough. But there is some kind of insects in it. So I don't know, in a way I would like to see some microbiology, definitely some parasites also shown in this. Then I also have been wondering about the selection of the animals in this image, because there are quite different types of animals shown. And where my attention kind of goes is the green chicken, because I am veterinarian as my background education, and I had never seen a green chicken, and I was wondering what this is. But and actually just recently I was visiting colleagues in Germany, and one colleague afterwards told me that there is a chicken breed that is basically the feathers, and it's like black, but in a certain light it can look green. So it's a real thing to have a very healthy green chicken. But of course, it's challenging to show what One Health is. I think one key thing is that it's an kind of a way of thinking, and we can all look at it and find our own point of views in it, and that's the beauty of it. And in this sense, we who are interested in parasitology and parasites, we are like in a good position, because parasites are really quite One Health. Some, of course, directly, if you talk about classical example of zoonotic parasites or parasites that have an environmental stage. But generally, I mean, parasites are special. We know that. And if you think about parasite life cycle illustrations, they very typically show some key connections between the different stages of the parasite, if it has that in a way, different hosts, and often also something about the environment. And if you think that it's very classical thinking in our head is that in addition to the characteristics of the parasites, it's also very important to their story, what is the health status of their hosts and the environment. They certainly shape the storyline. And here are just some thoughts on this, how parasites are really special and in this kind of One Health thinking. A diverse group of organisms, so we need to always to be able to understand different parasites. We cannot be in too much in one box. Typically, quite many with complex life cycles, with many hosts and many transmission ways and pathways that can be interconnected. Can be a wide host range or very limited host ranges. And also here comes the zoonotic parasites, of course, as a key area. Interactions between hosts can affect the parasites. Then, of course, how the parasite and the host interact, that is really interesting aspects there. Something that always discussing with people working with other types of organisms. We have the best stories in parasitology. Then all the concept of like hosting, being a parasite, coexisting, how the balance is that usually a good parasite would not be harming its host too much, so that they can live together as long as it's good for both, or at least the parasites. The host can be reservoirs. If it's a parasite that can cause disease, there can be subclinical infections and then clinical disease, and even can be causing death of the host, the infection. Then indeed, many parasites have an environmental stage, so there is really a link to that aspect of One Health, that generally in One Health discussions, it's said that integrating and involving the environmental sector is challenging. But I think in parasitology, we have good links and good importance, and there is relevance of that side. Parasites themselves are also part of the biodiversity, can have different roles in ecological and evolutionary processes. So that part of it's also very interesting to think about. And parasites are thriving across the globe in very different settings. So there are challenges and interesting stories both locally and globally. So kind of also the different stage of that were shown in the One Health illustration go very nicely with parasites. And I have actually been sometimes thinking of this like One Health, One Parasitology, how the parasitology actually is bringing in an interesting way, people from different disciplines and expertise areas together and across countries too. We are interconnected by different collaborations and our shared interests. So I mean, parasites are special also in that sense that they are bringing us together. And how we are, I think we are also special. We are curious, we are collaborative, we are sharing our views to kind of together then get a better understanding of how the different parasites have their life ongoing. So this sentence here, that parasitology gives opportunities to both practice and develop One Health ways of thinking and working is kind of my key conclusion, that we could teach parasitology in all the One Health education initiatives. And on the same topic, in the summer of 23, I was giving a keynote at the Congress of the Scandinavian Baltic Society for Parasitology, also linking to One Health. And that time, AI tools were quite new. And the version of ChatGPT available at that point, I was giving this publication with the One Health definition, and then a few ideas of like how could this link to parasitology to this tool. And this is the outcome, a short poem, how parasitology can be really a lens, like a way of looking and understanding life and One Health, and how those all things are intertwined and linked to each other. So just a part of, this is the cultural part of the talk. Then back to this picture. And to my example, about very general example, if you think about surveillance of different pathogens or different topics and diseases, this is related to my daily work. So surveillance of zoonotic pathogens, we can each think of as zoonotic parasites of our choice in our mind. And classically for surveillance, we talk about that there is this surveillance pyramid. If you think about humans that can get infected, that might get sick from it, that might need to be hospitalized or even die from the infection. And then it's kind of like there's the number of like how many there are in each layer, but also typically there are some gaps in the data. There might be not so good records from the hospital. Maybe there is only data on individuals that actually are really severely sick or die. Or there might be a lot of people who are infected but not tested, and we don't even know about them. So there's a lot of this kind of uncertainty across these different parts of the pyramid. But if you think this is a zoonotic parasite, so it's not this lonely the view, but there might be other host species that also host these same parasites, and maybe it causes disease in them. Some zoonotic parasites only cause disease in some of their hosts, but there might be still different pyramids. And if we would understand and have this overview how it's looking there, we might have a lot better general overview of the burden this zoonotic pathogen parasite is causing. Here, certainly gaps in the information might be very different across host-specific pyramids, but together and just kind of imagining the parts that are not there or modeling them, that might give a better idea. And then if we include the environments, because many parasites have the environmental stages, and if there are data available of their presence in the environment, that of course both links these pyramids very often, but also can give like additional information beyond what is available from the data sources that are classical involved. And this is then when it all gets very interesting. So here I've added an illustration of systems thinking on top. And there we would identify different entities and actions so that are across these different like structures in this view. And the point is there can be, for example, a hospital, there can be a laboratory, there can be a research group or something like that. And when these different entities start working together and sharing information. So if you talk about surveillance, then that would be integrated One Health surveillance and giving a better idea together. And if this surveillance aims for early detection, then the emergency of something new would appear in the system somewhere. The line there, the black line with the dots is kind of like where would something new is coming, where would it be noticed? But all this also goes for collaborations other than surveillance, so research and just discussions together. It's really good for general understanding of parasites to talk with people who are looking at it from different point of views and may have different data on it and so on. Because also we all have blind spots. We might think we know the parasites we have been working with, but maybe there's something new coming, and also good questions across the fields, because we sometimes forget to ask questions. Back to this picture. So now when we have looked at it from the surveillance part, I go to my second example, which is a One Health collaboration project focusing on source attribution. What are the sources of a parasitic infection? And this specific project focuses on a specific parasite, Toxoplasma gondii, which is a really good example for where One Health thinking is used and also needed. So the project is actually formally ended. It was funded on the One Health European Joint Program. Project is called Toxosources. But this consortium, we are still keeping in touch and working together and actually linking to other networks globally to continue the work on focus on Toxoplasma and indeed using One Health approach. This project was interesting because it was indeed part of a large European initiative, One Health research projects umbrella, One Health EJP, or European Joint Program. There were a number of projects focusing on parasites, but it was still a minority. But it was really nice to work across and together with colleagues focusing on other types of organisms like bacteriology and virology and epidemiology generally and so on. So also a nice way to just bring people together. I think One Health does that. Here is an illustration of basically kind of different sources or like pathways of human Toxoplasma gondii infection. Toxoplasma gondii is a protozoan parasite that has 3 different infective stages, and the infection can be acquired by many ways. Most of the infections in most host species are acquired after birth during the lifetime of the host. And here in this illustration, it's illustrated by the arrows that are going to the 2 adult humans. The congenital infection is also practically a further arrow from the acquired infection of the mother. And this is now focusing on humans and how humans, what are the sources of how humans might get Toxoplasma gondii infection. But if you look at it with the One Health point of view, we can see there's more in this illustration. If we still look at the humans, so to simplify these different arrows coming there, we can simplify them to be the meat-borne pathway. So the humans might be eating some meat that's or other tissues of infected animals that have tissue cysts of Toxoplasma gondii, and that can be a source of infection. And the other pathway is the environmental pathway. So ingesting oocysts that can have contaminated food or water or the environment. Practically, that environmental pathway is a fecal-oral transmission because these oocysts are the forms that are shed by infected cats, phelips, in their feces. If we think about the meat-borne pathway, those tissue cysts, they don't just appear there in these animals. They exist only if the animal is infected, and those infections are very often from oocysts. So going kind of upstream the arrows, very important to highlight also that the oocysts, they don't just appear. They are only there if the cat gets the infection, then it can shed. And this image is actually corrected a little bit because in the original, in the upper part, there is kind of the oocysts going from the small rodent and bird to the cats. And I would say it's more logical that it's the tissue cysts. And then there's the additional arrow in the upper corner is actually that very often for domestic cats is the owners of the cats who give raw meat to their pets can be also giving the infection to the cats. Back to focusing on the arrows going to humans, the relative contribution of these different arrows is largely unknown. and likely differs by setting across the globe, and even across Europe. And not knowing more about the source attribution, how can this be? We don't know where these infections are coming. It has been partly due to lack of methods that could be used, both in laboratory and modeling approaches, because there are source attribution methods for bacterial infections, for example, but really to use them for parasitic infections, they are not all directly applicable. And that's quite a gap, because Toxoplasma gondii is one of the top priority foodborne parasites, highly ranked both globally and in Europe, where this specific project is mainly set. And thus we got this funding for Toxosources to work on a question: what are the relative contributions of different sources of Toxoplasma gondii infection in humans, and really applying One Health approach and expertise in this. So key was multidisciplinary approaches, and also we were a large consortium. Here is a picture of those who joined the hybrid kickoff meeting on site, but we were more than 20 partners across Europe in this project. Multidisciplinary approaches, I would say that is also One Health way of thinking, like everybody can contribute with their expertise, and together we answer this quite simple sounding question then, or contribute to understanding that. And here are the key approaches we focused and key people mentioned, and we were building on previous work and collaborated also with other projects, and aim to take benefit of the large consortium. So there was a lot of multi-center studies and implementing methods in many laboratories. And I have just quick tasters of some of the examples of the work done in the different groups or topics on the next slides. So we focused on improving approach that has been used previously, but mainly for pathogens, quantitative microbiological risk assessment. And we did data for a multi-center QMRA for Toxoplasma gondii, and conducted literature reviews focusing on geographical differences that we were expecting will be there, and a large scale quantitative exposure survey, so questionnaire to human population based on experiences from the Netherlands, and we expanded it to also include the environmental pathway, because previously it was focusing on food that people are eating. And that was expanded to 9 countries. And the survey data analysis are ongoing, but very interesting overviews of the situation already available. Then we had a focus on fresh produce, because the eating habits are changing. Previously, Toxoplasma as foodborne pathogen was a lot of focus on the meat-borne pathway. And for this, we worked to select methods for how to detect oocyst contamination in different types of fresh produce. Again, literature reviews and step-by-step development of a standard operating procedure that was then implemented in several laboratories and followed by a ring trial. And the validated methods was then used in a multi-center study, so quite like robust step-by-step approach. And we focused on ready-to-eat salads, because we knew that there are some discussions about other types of foodborne pathogens in these kind of products, but parasites have been neglected or not really been in the focus. And we detected Toxoplasma gondii oocyst contamination based on a molecular method on 4.1% of the salad samples, and it was more than 3,000 samples collected and tested. So quite interesting new aspects. So maybe the next picture for the different sources will highlight, for example, this fresh produce differently. I need to highlight that that was a molecular methods and not a method to show viability of the oocysts, but they are really robust, so we can interpret or consider that it's possible that there is infective oocyst contamination. We also worked on exploring serology for detecting Toxoplasma gondii infections caused by oocysts, because this would be brilliant for the understanding of proportions and so on. Again, review, and we prepared a workflow how to develop an assay to do this, to differentiate oocyst-driven infections from tissue cyst-driven infections. And this was really done from a One Health perspective also, because this is not only relevant to humans, but can also be very relevant for some omnivorous animal species that could guide really interventions later. So a step-by-step approach, starting with bioinformatics selection of proteins of interest, and then we used very well characterized reference sera from experimentally infected animals, and kind of build on this understanding and being critical, because there are some proteins described in the literature that have appeared promising for this use. But conclusion from the work that we did under toxosources thus far is that currently there's no antigen that would allow robust estimates of what proportion of Toxoplasma gondii infections have been acquired from oocysts by serology. But some really interesting insights that can guide further research and developments, and certainly a One Health issue. Then we also had focus on genotyping in this, yet another totally different expertise area brought to this. We worked to harmonize microsatellite typing that is used for both human and animal samples across Europe, and developed a new tool, typing tool, that is better fit for the European needs, because in Europe the strains circulating are very similar. And for this we also did a major work to get done some whole genome sequencing of a collection of European Toxoplasma gondii strains, to really find those polymorphic regions that can be detected in closely related strains. But again, something that is different in different parts of the world, the needs, and something that is relevant across typing samples from not only humans, but also animals and possibly also environments. So that was the last of the approaches in this project. And yeah, during this toxosources collaborations, I mean, of course, there's a major advancements of the research questions and new data of the different aspects. And but I would say it's just the consortium and the way of working. That's a major outcome that I'm so happy to have been part of and leading the work. So really a take-home message from this example is that based on this project, and this is my very biased view, but really a strength to have a consortium that is cross-sectoral, international, different expertise areas working together in a One Health collaboration to bring different knowledge together to address the research questions about parasites that really require One Health approach. And also the questions sometimes are really surprising. They are quite simple questions that we don't know if we only would be working in one area of it. And additionally, also one key thing is that often the teams in parasitology are quite small in the different organizations and countries, and working together you get this critical mass of working together and sharing, and that is the One Health way to do it. So indeed, it's a lot of different examples can go somewhere in this. I mean, it doesn't show the food. There's no salads in this picture, but they are, of course, they're somehow linked to each other. And but it goes really to this idea that One Health is an approach that can be used in collaborations where we need to work across expertise areas and disciplines and sectors and countries. And yeah, bringing it all together. Parasites are special. They can really be the core of really nice One Health collaborations, where we can also maybe show to other fields how it's done and what are the challenges, but also the success stories of doing it the One Health way. And this is my final key messages. again, One Health, One Parasitology, the idea that parasitology and the different work we each do, they can have interesting opportunities to work in a One Health way and develop also the One Health way of thinking. And I encourage to discuss outside the normal kind of network you might have and look into some new aspects because that's really where it's the added value comes from. So that's the key thing, working together with One Health approach when relevant. And right now, our direction is working together towards ICOPA 2026. And that's all from me. Thank you so much.